All right, so the next session is presentation of cutting-edge research, state-of-the-art research by some of the internationally renowned researchers who are here. In my first few intro remarks, I want to justify why the executive committee chose these four areas. So, as we discussed earlier, we have four overarching themes: TDP-43 as a non-amyloid mechanism and generalizability. And some of the rationale why we went for TDP-43 are here. There's, as I mentioned early on, that there's converging evidence from autopsy series and other studies that TDP-43 is an integral co-occurring pathology among many neurodegenerative diseases, including Alzheimer's disease. And it plays a major role in gene expression and is involved with the RNA metabolism and synthesis. On autopsy series, it is seen in about up to 60% of decedents who have a diagnosis of Alzheimer's dementia. Also, it is seen in other neurodegenerative diseases, including the Lewy body dementia, Parkinson's disease, Huntington, corticobasal degeneration, and so on. And finally, we know that TDP-43 contributes to the clinical phenotype, the psychiatric, cognitive, and other symptoms that are seen in patients with neurodegenerative diseases. So this pathology also has a role in clinical symptomatology of various diseases. It is a primary proteinopathy in FTD, LD, ALS, and late. And significant advances have been made regarding TDP-43 about its genetics, the structure, and the molecular mechanisms that may lie in the propagation of this protein. It moves from the nucleus to the cytoplasm and back, and we have a better understanding that it has a prion-like movement, and what are the molecular mechanisms underlying those movements, and perhaps we may have some discussion about it here today. Also, there's some new therapies that are being developed right now, and there may be some promising therapies coming up targeting TDP-43. So these are multiple reasons why we chose TDP-43 as one of the themes. And Dr. Margaret Flanagan will talk about the basic biology of TDP-43. I'll introduce her very shortly. And then another presentation by Dr. David Vogt, who will focus on the clinical definitions and biomarker development for late. And the other area, I'm sorry about the slide is busy, but just to make a case, and we've talked a lot about generalizability, how critical it is for our field. And it is a bit of a limitation right now in the research that we do, but we have seen major movement in enhancing generalizability in a big way by diversifying our cohorts and some other ways. The other way is the real-world data and the real-world evidence research, which Dr. Califf will talk about soon. So enhancing generalizability is one of the priorities for us, and certainly the diversity will help us examine the effects of genetics, lifestyle, environmental factors on risk and resilience to ADRD. As we diversify our cohorts, this is some of the major expansion we'll see in our research. And there are some limitations in terms of diversifying our cohorts. We have talked a lot about it. I will not dwell on this, but one of the things talked about in literature is the selection bias. Most of the ADRCs, the cohorts we recruit, there's a selection bias because the people whom we recruit, number one, it's a non-random selection of those participants, and they usually come to us. They come to our specialty clinics. If we don't go to the community to recruit them, they come to us. So there's already a bias in the kind of participants who come to the ADRCs and the specialty clinics. So that's the selection bias. And of course, there's another bias of difference in survivability between the URG and non-URG participants. So there's a survival bias and a selection bias that we deal with all the time and face. And these needs to be addressed. It is clear that real-world data research and real-world evidence will significantly enhance the generalizability of our research. These are the data that are collected at the point of care. This is when the patients go to the clinic, you see them, you collect the data, and these data are most relevant to patient care because you're collecting them as the patients report to them, and you examine them and do all kinds of tests in the clinic. So using real-world data research, especially through electronic health record, has many advantages, and some of them are here on the slide. It will help us identify a cohort that will enhance AD research. It will help us examine various risk factors that you will collect that information from the electronic health record and expand the field. And also, you can calculate time to event outcomes. And one of them I was thinking is the transition of patients from preclinical to clinical stages of the disease. And you see these people in your clinics, you could calculate the time of conversion just based on the electronic health record. Of course, the biomarker data and all those will help as well. So there are multiple advantages of doing real-world data research using electronic health record, and Dr. Califf will talk about it. So in the generalizability area, we'll have two presentations, one by Dr. Rachel Whitmer from UC Davis. She'll focus on selection bias and representation, which we talked early on, and Dr. Califf will talk about the real-world data with a focus on electronic health record. So with that, I would like to invite our first speaker, Dr. Margaret Flanagan. She's going to talk about the basic biology of TDP-43. Dr. Flanagan is a neuropathologist whose research focuses on the pathobiology of Alzheimer's disease and frontotemporal dementia. She is an associate professor of pathology at UT San Antonio and is also the neuropath core leader at South Texas ADRC. She is also co-director of the Brain Bank at the Biggs Institute at UT Health San Antonio. The primary focus of her research relates to the neurobiology of TDP-43 and its role in the development of cognitive impairment and dementia. Please help me welcome Dr. Flanagan to make a presentation. Dr. Flanagan.
Thank you very much for the introduction, and thank you for the opportunity to present today. Welcome to Texas, everyone. Today I'll be talking about the basic biology of TDP-43, which is tough to do in the allowed time that I was given, but I'll do my best. I have no conflicts to disclose. So as we know here, dementia very commonly is associated with the presence of multiple coexisting pathologies in the brain. And today I'll be focusing on TDP proteinopathies, so FTLD, TDP, and late neuropathologic change. And I'll briefly touch on some of the coexisting Alzheimer's disease, so amyloid plaques with TDP-43, as well as neurofibrillary tangles with TDP-43. So as we know, it has a lot of clinical prevalence. I won't spend too much time on this. A lot of individuals in the audience have shown in their work that there's a higher likelihood of dementia when beyond the amygdala stage, and associations have been described with hippocampal sclerosis of aging, tau tangle density, and amyloid beta burden. TDP-43 is a highly conserved heterogeneous nuclear ribonucleoprotein that's located on chromosome 1 and encodes a 414 amino acid protein. The TDP gene is essential in mammals, zebrafish, and flies, but interestingly, it's not essential in C. elegans. It's broadly expressed, and its expression is very tightly regulated, and it directly regulates its own expression and promotes its degradation. And why is it important to understand what TDP normally does? Well, this is because understanding TDP's normal structure, function, and pathologic aberrations are absolutely crucial to be able to delineate and effectively pharmacologically target mechanisms of disease in dementia patients. So structure and function. TDP-43 has high specificity for UG-rich RNA and TG-rich DNA. It has an N-terminal domain shown in the top in red, in A, and it has ubiquitin-like fold in the N-terminal structural domain, which is highlighted in B. It has also in the N-terminal domain a canonical nuclear localization sequence, and the N-terminal domain is responsible for the regulation of TDP homodimerization to ensure appropriate folding and mRNA splicing. In addition, TDP43 has 2 RNA recognition motifs, or RRMs, and I would just like to highlight in orange in A, RRM2, which specifically contains nuclear export sequence, which I'll elaborate on in subsequent slides. Then in green in A, there's a C-terminal domain, or a prion-like domain, which is low complexity and particularly prone to TDP aggregation and TDP inclusion formation. It's important for mRNA splicing and HN-RMP interactions, in addition to being the site of over 50 sporadic and ALS-associated mutations. In the C-terminal domain, it is an amyloidogenic core region with 2 alpha helices that convert into beta sheets in TDP43 aggregates. Next, moving on to the canonical nuclear localization sequence, which is specifically critical for TDP's physiologic function. Mutations in the nuclear localization sequence, shown in dark blue in the figure, have been shown to result in mislocalization and aggregation of TDP43. And because we know this, we are able to manipulate NLS in order to create better model systems that are more characteristic of what we see in the human brain during disease to improve our models. So I'll briefly show an example of this in the next slide. So here I'd just briefly like to show a new Alzheimer's disease TDP43 late neuropathologic change mouse model that I've been working on in collaboration with Dr. Michael Gitcho in Delaware. And for this mouse model, we selectively express human TDP43 and TDP with a defective nuclear localization signal in the hippocampus and amyloid APP PSCN1 background. And these 24-month-old mice display severe neuronal loss in the hippocampus, as shown here in the figures, which, as you can see, very closely recapitulates what we see in the human brain in hippocampal sclerosis of aging, which is very strongly associated with the presence of pathologic TDP43 in both late as well as FTLD TDP43. In addition, this mouse shows changes in amyloid beta deposition, shown at the bottom of this slide, specifically decrease in amyloid beta plaque counts, as well as variability in amyloid plaque size. And our new hippocampal sclerosis-like model may ultimately provide a greater understanding of the pathogenesis of neurodegeneration seen in TDP proteinopathies, particularly in the presence of coexisting amyloid beta, which is quite common. Now to elaborate on the nuclear export sequence, it remains somewhat controversial, although appears to be becoming more clear. So as we know, TDP is exported from the nucleus to the cytoplasm, and this had been thought to be mediated by exportin XPO1 binding to the nuclear export sequence in RRM2, as shown in orange again. But in addition, more recent data actually suggests that the export of TDP from the nucleus to the cytoplasm does not require XPO1 or the nuclear export sequence, and perhaps it may be due to passive diffusion. So more work is necessary to clarify. So the function of TDP, it's important for the regulation of gene expression and other aspects of RNA processing. And it targets over 4,000 different mRNA transcripts that range from disease-associated transcripts to its own mRNA. An example is that TDP may downregulate tau expression by destabilizing mRNA transcripts and may regulate the ratio of 3R to 4R tau by alternative splicing of tau exon 10. In another study, however, it was not replicated, so again, further work is necessary in this area to clarify. Shuttling of TDP between the cytoplasm and nucleus is dependent on transcriptional needs, and levels of TDP43 is altered in human neurons, particularly in disease settings of FTLD, TDP, and ALS. And in the disease settings, higher amounts of the mitochondrial TDP alters morphology and impairs mitochondrial function. So in summary, TDP plays pivotal roles in alternative splicing, transcriptional regulation, mRNA stabilization, and is involved in the cellular response. So when a cell is exposed to stressors, it regulates the TDP regulates mRNA levels to specifically conserve energy and prioritize the cell's survival. TDP can associate with ribosomes and stress granules to temporarily stop translation and promote cytoprotective protein synthesis. So to briefly just touch on TDP's relationship with tau, just a couple of key publications here. So overexpression of phosphorylated TDP in APP-PSCN1 mutation transgenic Alzheimer mouse model demonstrated increased hyperphosphorylated tau immunoreactivity. Physiologic TDP may specifically have a suppressive role in the regulation of tau mRNA, and when TDP's function is lost, there are increases in tau mRNA production and subsequent translocation of tau proteins become available for hyperphosphorylation. And then I just wanted to highlight in publication from the Mayo Group that they described the type beta inclusions in the human brain to be associated with tau neurofibrillary tangles in the same neuron, particularly in the hippocampal and entorhinal regions, highlighting the relevance of studying this in more detail because of the relevance to human disease. And then the figure at the bottom is a study from McAleese et al. That compared Alzheimer's disease by itself with Alzheimer's disease with TDP-43 and quantified the amount of tau, both early with MC1 as well as later phosphorylated tau in numerous brain regions, and they found no significant difference between groups. To touch on TDP's relationship with amyloid beta, again, some key findings have been that knocking out TDP in the hippocampus and cortex of an APP background reduced amyloid beta plaques while accelerating neurodegeneration and leading to cognitive deficits. This is related to cryptic exons specifically, and then additionally it's been reported that the nuclear depletion of TDP-43 in the forebrain neurons of an 80 mouse model exacerbated degeneration and correlated with increased prefibrillar oligomeric A beta and decreased plaque burden. From the same paper that I talked about in the previous slide, they also looked at differences in the setting of AD with and without TDP-43 with late NC, and also found no significant difference. So taken together, we really need to further investigate the relationships of TDP with both amyloid beta and tau for further clarification. I don't have time to go into this, but I included this table just to refer back to, and I'm going to be sharing my slides. This is from a recent paper from Pete Nelson summarizing genes linked to late NC hippocampal sclerosis, neuropathologic phenotypes. The top of the table are genes that have been replicated, the bottom not yet replicated, with disease-related phenotypes in the middle column, and additional selected references for further reading. So interestingly, more recently in the TDP field, there's been increased interest in TMEM106B. TMEM106B is a gene on chromosome 7 that encodes for a protein that is highest or expressed most highly in oligodendrocytes and neurons. And we know that variants in this gene can affect the risk and severity of frontotemporal dementia. There's both a protective variant that inhibits filament formation and delays disease progression, and a risk variant that promotes filament formation, accelerating disease and the progression. In a recent workshop that was held on TDP, TMEM106B was beautifully presented by Dr. Ian McKenzie. So just to kind of summarize his talk very quickly, there were kind of these 3 papers that came out. 2022, the first found that these filaments were consistently found in 4 FTLD-GRN cases, as well as an older progressive supranuclear palsy case. So these findings raise the question, could TMEM106B filaments be an age-related presence and seen in various conditions? Another paper identified TMEM106B as a filamentous protein in other types of FTLD-TDP, specifically challenging the exclusive focus of the field on TDP-43 specifically. And then the third looked at 25 brain samples across various diseases and ages, and findings from this study suggest the filaments may actually be an age-related feature and raise the question, are they potentially independent of specific diseases? So in summary, accumulation of C-terminal fragments and filaments of TMEM106B, it's a common age-related process. TMEM106B is subcellular and important in lysosomal function, so it may also have a high potential for a lysosome-specific biomarker. And there have been consistent features described in FTD, GRN, as well as late NC. It may be a potential therapeutic target, or perhaps only certain species of TMEM106B C-terminal fragments may be related to disease states. So pathologic TDP has two things simultaneously happening: loss of normal nuclear localization and cytoplasmic protein aggregation. So what happens when we lose normal nuclear function? The nuclear depletion leads to loss of normal RNA regulatory functions. There's disruption of RNA metabolism, leading to widespread dysregulation of RNA species that TDP regulates. You may also have impairment of the neuron's ability to cope with stress and repair mechanisms, and neurons become more vulnerable to other toxic processes, including those that are triggered by the accumulation of TDP inclusions in the cytoplasm themselves. Therefore, therapeutic strategies to enhance TDP nuclear localization are critical. And because TDP nuclear depletion is a key feature of its pathology, some of these approaches may include modulating nuclear transport receptors or inhibiting nuclear export signals to retain TDP in the nucleus. So now to touch on the cytoplasmic protein aggregation. Again, the C-domain prion-like domain plays a critical role as it promotes misfolding into toxic oligomers and fibrillar aggregates. Additionally, post-translational modifications impair TDP's RNA binding and splicing. This includes phosphorylation and acetylation, and TDP is recruited to stress granules under cellular stress. Some therapeutic strategies to specifically target the aggregation of TDP include inhibition of aggregation, such as using small molecules to stable TDP's native conformation, disaggregation of existing inclusions, gene therapy, such as reducing mutant TDP or modifying the post-translational processing, and RNA interference strategies to knock down specific genetic contributors to pathologic TDP43. So in conclusion, the basic biology is very complex and multifaceted for TDP, even by itself, but then when you consider coexisting amyloid beta or tau or alpha-synuclein, which I didn't have time to touch on today, it gets even more complicated. There's also the emerging role of TMEM106B, and the loss of function of TDP is particularly insidious as it's less visible than the gain of toxic function, but potentially just as harmful. Therefore, for therapeutic strategies, we must aim not only to prevent toxic gain of function, for example, the aggregation mislocalization, but we must simultaneously aim to restore or compensate for the loss of the TDP normal nuclear function. So a dual approach, or even more simultaneous targeted approaches, will become essential for effectively addressing the multifaceted nature of TDP's pathobiology and neurodegenerative disease in the human brain. So I have a comprehensive reference list because that was a very quick overview, and yeah, thank you.
Thank you, Dr. Flanagan. We'll have a Q&A session after the next presentation. So the next presenter is Dr. David Vogt. Dr. Vogt is the director of Penn ADRC. He's chief of the Division of Cognitive Neurology and a member of the NAC Steering Committee. He also co-directs the Penn Institute on Aging and Memory Center. His research focuses on the cognitive neuroscience and memory decline in aging and Alzheimer's disease, and his studies employ multiple state-of-the-art technologies, including MR, FDG, and molecular PET imaging. Another area of his research interest relates to identifying heterogeneity, which is seen in Alzheimer's disease. Dr. Vogt has received continuous NIH funding for the last 20 years, and he has been the PI or a co-I of multiple national and international studies. Please help me welcoming Dr. Vogt to make his presentation.
Thanks so much, everyone, and really thanks for the invitation. It's a great honor to speak here today. So I'm going to talk a little bit about how we can begin to detect clinically late, and I'll also describe some new clinical criteria that we're proposing as a possibility for detection. Here are my disclosures. So why should we care about detecting late NC clinically? I hope I don't have to convince too many of you of the importance of this, but there's an increased awareness, obviously, that TDP-43 is an important modulator of late life cognitive decline. And I think with the reification of a pathologic criteria for late, led by Pete Nelson and others, that's really kind of accelerated the recognition of this pathologic entity. And with this increased awareness, I think what's happening is in our clinics, I think we're starting to see patients that we think, this might be someone who has TDP-43 associated memory decline. And I would say that this importance is even further amplified with the emergence of disease-modifying medicines targeting AD, because late really is a mimic of AD, and so many of the patients we're working up for drugs like lecanemab, who are amyloid negative, will have late as their underlying pathology. And late also, as was just discussed, frequently co-occurs with Alzheimer's disease, and we really don't know much about how that impacts the outcomes with these drugs. The idea that TDP-43 is common in older age and has some cognitive effect is not really a new one. So back in 1994, Dennis Dixon described hippocampal sclerosis occurring in about 25% of people over the age of 80, and he described it as a amnestic syndrome that was disproportionate to other cognitive deficits. And then shortly after the discovery of TDP-43, it was found to be really very commonly associated with hippocampal sclerosis, probably in 90% or more of these individuals. The next decade after, there was quite a bit of work looking at TDP-43 and aging. A lot of that was really done by Keith Josephs at Mayo, where he showed that both TDP-43 and AD independently are associated with changes in hippocampal structure and memory decline, and independent of hippocampal sclerosis as well. So you didn't need to have hippocampal sclerosis to also have memory decline in the setting of TDP-43. And in general, what was found in a number of studies done by a variety of people here in this room is that even in individuals who don't have Alzheimer's disease or have low levels of Alzheimer's disease neuropathic change, more than a third of individuals over the age of 80 have some degree of late NC, and it's associated with memory deficits. And regardless of whether it occurs with Alzheimer's disease or more as a sort of primary pathology, it tends to affect limbic structures in a relatively stereotyped pattern. And so that led to this designation or definition of a pathologic entity of late NC, which was originally put out in 2019 and updated to sort of simplify the staging in 2023. And the staging, as you can see on your left, that is part of these new criteria, is relatively simplified. Stage 1, there's amygdala involvement. Stage 2, hippocampus. And then there's movement into the middle frontal gyrus. But actually, prior to this staging scheme, there were a couple other staging schemes out there, one developed by Keith Josephs, another by Julie Schneider's group, which I think tell us a little bit about more of the anatomy of what regions are affected with TDP-43, and perhaps what we might see in terms of structural imaging and other forms of spatial neurodegenerative markers. And so, for example, there's involvement of the insular cortex, involvement of the temporal pole, and orbital frontal cortex, which may influence the presentation. But of course, this is an entity that strongly affects the hippocampus, and when looking across domains, individuals with TDP-43 have relatively selective amnestic syndromes. Episodic memory is the domain most affected. They tend to have a relatively indolent course, so compared to Alzheimer's disease, this is a slower moving entity when it is the sort of primary pathology. Occasionally, I think to variable degrees, they may have some degree of semantic memory impairment as well, perhaps related to the temporal polar involvement, often measured by things like category fluency. And they do have some degree of, depending on who you talk to, but I think we see patients who have some neurobehavioral symptoms associated with this, not to the degree of typical behavioral variant FTLD, but certainly some degree of disinhibition and other behavioral changes. And I do think, there's arguments for thinking of these things on more of a continuum. When late NC occurs with hippocampal sclerosis, it tends to be more aggressive in terms of the degree of cognitive decline. So this is a study done by the Rush group in which they looked across different domains. And in Alzheimer's disease, you see over time the typical amnestic multi-domain syndrome with episodic memory as early and prominent. With TDP-43 alone, episodic memory is most affected, but in the setting of hippocampal sclerosis, the degree of memory impairment can be quite severe and relatively rapid, and there often is involvement of semantic memory. Of course, late NC really commonly occurs in the setting of Alzheimer's disease, as was mentioned earlier. And so this is work from Pete Nelson, where they looked at 13 community and population-based autopsy cohorts, so a really large set of patients. And what was found were a couple things. One is that the severity of Alzheimer's disease, based on plaques and tangles, was associated with higher rates of TDP-43 copathology, and more than half of the people with late or severe AD pathology also had late NC. And perhaps not surprisingly, but important for our patients, when these conditions are mixed, now you see a much more rapid rate of decline cognitively in these individuals relative to typical Alzheimer's disease. Well, one would expect, given the topography of where TDP-43 is in the brain, that it would affect hippocampal volumes, and indeed it does. This is postmortem imaging from the ROSMAP group, in which they looked at several different pathologies in terms of hippocampal volume. And if you look at the AD group, you see some decrease in hippocampal volume relative to people without pathology. But in the late group and in the AD plus late group, there's much more severe hippocampal atrophy on average. And if you kind of think about the full distribution of AD across these AD and AD plus late groups, and you kind of look at the bottom quartile of individuals, what you see is that, when you get into the lower range of what we think of as the continuum of AD hippocampal atrophy, those cases tend to be enriched in people with late, whereas when there's less atrophy, you tend to see less late. In addition to these cross-sectional measures, there's data again from Keith Joseph's group showing a more rapid rate of hippocampal atrophy over time in those individuals with Alzheimer's with TDP-43 versus those without. When we look cortically, we see a pattern that really matches the staging that I mentioned earlier. This is again on the your left, the postmortem imaging again of individuals with late, and the pattern of areas in which there is cortical atrophy. And this pattern, the sort of hottest spots are in medial temporal lobe, temporal pole, and orbital frontal cortex. And then on the right is images that are from the Mayo group, in which they had antemortem MRI imaging in individuals who had TDP-43. And what you see again is a kind of gradient of atrophy cortically in anterior medial temporal lobe and into the temporal pole and a little bit in frontoinsular areas. So again, kind of matching the pattern of where TDP-43 is in the brain. We also have observed this gradient of anterior to posterior within the medial temporal lobe itself. And so this is a study we did in patients with either intermediate or high levels of ADNC who we had antemortem imaging. And when you look at tau pathology and its relationship to atrophy, there's a relationship kind of along the long axis of the hippocampus, which has been described by others. There's not necessarily a preference along the hippocampus, whereas with TDP-43, we see much greater atrophy in the head of the hippocampus, as well as anterior structures in like anterior entorhinal cortex. And in fact, if you take kind of a ratio of anterior structures to posterior structures, we got pretty good discrimination between those with and without TDP-43 who have Alzheimer's disease concomitantly. FDG PET has also proven to be, I think, a pretty useful neurodegenerative marker for late. A lot of this work has been championed by David Jones at Mayo Clinic, and his group have made the observation that in late, relative to Alzheimer's disease, these individuals have greater hypometabolism, particularly in the medial temporal lobe, so more so than typical Alzheimer's, and also involve orbital frontal cortex, whereas Alzheimer's patients tend to have more hypometabolism in inferior and lateral temporal cortex. And so again, making a ratio of these areas produced pretty good discrimination between AD and late. And then recently, Michael Groth did, I think, a really clever study where he looked at patients from ADNI who had a diagnosis of clinical Alzheimer's disease and then took their FDG PET patterns and in a sort of similarity matching algorithm, found those that had more AD-like and those who had more late-like FDG PET. And those that were more late-like tended to have less abnormal AD biomarkers, less ApoE4, and higher actually TMEM106B, which is a genetic risk factor that we talked about, Dr. Flanagan just talked about. We've taken a sort of different approach to think about co-pathologies in Alzheimer's disease, a sort of a residual approach, in which we make this assumption that the majority of the variance in Alzheimer's disease is driven by tau pathology, to at least a first approximation, and we base this on tau PET. And we looked across the brain in all the regions in annotation of the entire cortex, in which we looked at the relationship on the y-axis between cortical thickness and on the x-axis tau PET SUVR. And we see a linear relationship, but we also see individuals who have, at the top, that have less atrophy than you'd expect for a given level of tau, so perhaps they have brain reserve or are more resilient. And then we see patients, though, that have very significant atrophy relative to their level of tau, and they may be more vulnerable. And we expect that some of them will have co-pathologies. And when we did this and looked at individual cases, we tended to see that there was a group of patients that had more temporal areas in which they had greater atrophy than expected for tau. We then went on and looked at residual maps of the individuals that we studied using both FDG PET and MRI. And we clustered these residual maps. So these are maps of people who we've regressed out Alzheimer's disease, if you will. And in both cases, we found limbic vulnerable individuals, individuals who had greater atrophy than expected in these regions. These groups who are mixed AD, we would hypothesize are mixed AD and late, have a faster rate of decline than a canonical group, the right amount of atrophy for tau. The pattern structurally looks quite a bit actually like the pattern that we saw in the postmortem imaging, and actually looks similar to amyloid negative symptomatic patients who we also clustered on cortical thickness. And then in an antemortem postmortem study, we also found these limbic vulnerable groups tended to have more TDP-43. So, smaller data set there, but interesting. There is actually a great deal of exciting work for biofluid biomarkers for TDP-43, so a pair of papers, one from Phil Wong's group at Hopkins and one from Mike Ward at NIA, have looked at these so-called cryptic peptides, which are based on the loss of function that Dr. Flanagan discussed earlier, where there's missplicing of RNA that leads to cryptic peptides, which in some cases are measurable. And these cryptic peptides seem to be associated with TDP-43 conditions. And so in particular, this one particular cryptic peptide that was described by Phil Wong's group was seen to be elevated in people with C9orf mutations pre-symptomatically, as well as sporadic ALS. And what was, I think, compelling is the relationship between these measures, both in CSF, was highly correlated to what was found in blood, which at least gives some reason to believe maybe we'll find blood-based biomarkers. There's been other blood-based work. This is from Robert Rissman's lab, and a couple other groups have looked at extracellular vesicles. In this particular analysis, they were looking at late cases versus those without, and in astrocytically derived extracellular vesicles, there were higher rates of TDP. There is some tracer work. I'm an imager. I prefer imaging over biofluids, but you know. So there has been some tracer work. AC Immune has a tracer that seems to be relatively selective for TDP type A. And this was generously shared by Tamara Sarandinina. And they're going to have first-in-human studies, my understanding is in the fourth quarter of this year. So we don't have a molecular marker for TDP-43. Why would we be so audacious to try to propose clinical criteria? Well, we're audacious people, I think. I've sort of said many of these. I think some of us are already making the diagnosis. The absence of a diagnosis, I think, impedes our clinical and basic research into pathogenesis and treatment. And it has different implications for our patients. And people who have primarily late as a disease-driving entity, they tend to have a slower course, which I think is useful information for people. And again, I do think in the context of FDA approval of anti-amyloid therapies, we're going to be diagnosing these individuals or finding individuals who are negative for amyloid PET or amyloid in CSF, and we're going to want to diagnose them with something. They're going to want to know what they have. And again, it's also unclear what this co-pathology, its implications are for treatment in AD. So we, a committee of 36 of us, were involved in a Gaps and Opportunities conference sponsored by NIA. This helped us develop a framework for diagnostic criteria. We realized that in the absence of a TDP-43 specific biomarker, our diagnosis is going to be at best probabilistic. But you know what? Our field's been very used to probabilistic diagnoses for a long time. And we defined sort of two primary contexts for late for diagnosis. One is when late NC is the primary driver and ADNC is thought to be less significant. And for those, we can call them possible and probable late. And the other is when ADNC is mixed with late. And at best, I feel like we could say possible late currently. We defined core clinical syndromes for each of these cases. Not surprisingly, for late as the disease-driving entity, we defined a primarily amnestic syndrome that was indolent, generally in individuals over the age of 75. And in mixed disease, it really is, typical of Alzheimer's disease and being an amnestic multi-domain syndrome, but we expect memory loss to be more severe relative to other domains and the course to be more rapid. And just to give you a very high overview of the criteria that we proposed, if someone has those core clinical criteria for late as a primary driver, they have to also have significant hippocampal atrophy. And if no amyloid biomarkers are available, at best we would say these are possible late. If they are available and they're amyloid negative, we think these cases are very likely to be late, particularly with significant hippocampal atrophy. Whereas if they're amyloid positive, they still could have late, but you need tau markers. And if they're tau negative, we say they have possible late, in part because of some concern about the sensitivity of our tau PET measures for medial temporal tau. Whereas if they're tau positive, they certainly have ADNC, they have amyloid and tau, but perhaps also could have mixed late. If they have mixed late in AD, they'll have the core clinical features we mentioned, but they're going to require other supportive features, such as hippocampal atrophy at a proportion to tau pathology or the level of impairment, as well as that FTG PET ratio. So I'm not going to read through all of these for the conclusion for time, but basically I think we can come up with probabilistic criteria at this point. All of this needs to be validated, though, and I think this is a good starting place for us to begin to validate these criteria. And so I just want to thank my collaborators and actually all the individuals who are involved in developing the clinical criteria that we worked on. Thank you.
Time for one or two questions, anyone?
Great session. I have a question for you, Dr. Flanagan. First of all, great presentation. I was just at a meeting, and in the spirit of multiple proteinopathies, I was just at a meeting that was wondering, can they all be coming from one mechanism, or is it just sort of separate and parallel, all the different proteinopathies? And in that particular meeting, someone was making the case that, they all seem, whether it's amyloid tau, alpha-synuclein, TMEM, TDP, they all seem to potentially emerge from endolysosomal dysfunction. So I was wondering if you can comment on that. I mean, I know you had one slide which indicated that, and I know there's a debate about, where do you place the lesion, so to speak, cellularly? Is it the nucleus or is it the cytosol? And, Len Petrocelli's beautiful work showing that it's truly the endolysosomal dysfunction that could trigger TDP-43. So, as you say, complicated story. Is there a possibility of simplification?
Yeah, great question. I hope so. So I guess in my opinion, I think it's still very much an emerging area and more research needs to be done, but I do think that there is a lot of overlap specifically for alpha-synuclein tau and TDP-43. I think amyloid beta and its nature being extracellular, I don't see as much overlap specifically involving lysosomal dysfunction, but as you mentioned, I think for the other three, it's more likely.
Yeah, thank you.
I think we'll move to the next session. Thank you so much. Thank you. Okay, our next presentation is by Dr. Rachel Whitmer. Dr. Whitmer is the co-director of the UC Davis ADRC. She's professor and vice chair for research and chief of the Division of Epidemiology at UC Davis Department of Public Health and Neurology. Her NIH-funded research focuses on identifying modifiable risk factors for brain health and dementia, especially in diverse populations that have been underrepresented in ADRC research. Please help me welcome Dr. Rachel Whitmer to make our presentation. Thank you.
Thank you so much, and thank you for this opportunity to talk about something I feel very deeply about, and it's on the list of things that keeps me awake at night. I don't have anything to disclose in reference to this talk. I do receive funding from the NIH and the Alzheimer's Association, and I have a Cypress Award. So I was asked to talk about representation and selection bias. And I just want everyone to sit for a moment and think about, what does representation actually mean? In our work at our centers, are we supposed to be representing a local population? If we do that, are we truly serving those who are not only underrepresented, but maybe most need to be included in this work? And should our approach be to engage those who are potentially at highest risk? And does that mean representation, and what does that look like? So I want to come away with some ideas about how do we broaden our scope to include all that are in underserved populations. How do we engage these groups in research, and what works and for whom, and what are the challenges and the resources needed? So when we're talking about selection bias and representation, you can't really talk about one without the other. If you want good representation, you try to avoid some type of sampling or selection bias, which of course in reality is not 100% unavoidable. And this is inextricably linked to our ability to do recruitment science, and it really is a science. So inclusion is an actual science. And this is some work that was led by Dr. Miguel Renteria that looked very closely at comparing those who are enrolled in centers with HRS, which is a study based on purposely stratified population-based sampling, and found that there were differences in those who are in the ADRC centers versus the HRS. And there were differences across different socio-demographic groups in terms of depression, education, subjective cognition, but also those differences were particularly augmented for those who are not non-Hispanic white. So it was particularly represented in those who are underrepresented in research. So you don't know what you don't know. You have to remember that the characteristics of people who opt out of participating in a survey are not studied. You can look at who perhaps came to an event, who showed interest. You can look at those who enrolled in your study but decided not to stay in your study, or perhaps decided not to do additional ancillary items like neuroimaging or a blood draw, but you can't get at the people even before that. We don't really know what are the characteristics of the people who decide not to participate at all. And this is where excellent qualitative work comes in and focus groups. There was some wonderful work led by Dr. Mary Ganguly, who really honed in on, within an existing cohort study, who wanted to participate in neuroimaging. And I suggest everyone to take a look at the study. It's from 2015, but it is very germane for today. Now, there's different things that one can do once you're in a study to try to look at things like sampling bias, selection bias, and what are the predictors of staying in a study. And this is just showing you a figure of our Life After 90 study, which is a study following everyone who starts off at age 90 or above. This study's been going on for about 6 years. They're all long-term members of Kaiser Northern California, and they have information from health data checkups from the 1960s and the 1970s. So it gives us a unique opportunity to actually look at who was there in the 1960s, who was part of this multiphasic health data collection, who then subsequently went on to die after that, who was available in 1996 when electronic records became available, and then who was eligible for the study when the study started in 2018, who met all this criteria but did not decide to enroll in the study. And you can actually calculate an inverse probability weight at each of these time points and use those weights to look at your conclusions in your work and understand how much has selection bias and survival bias and attrition bias affected the results and the associations that one is seeing. So when I was very early in my career, I think, I guess 25 years ago, when I was writing my first R01, one of the critiques that came back is what was I going to do about survival bias? I was proposing a cohort study at Kaiser, everyone age 65 plus, and I thought, well, what am I going to do? And I had a conversation with one of the program project officers, and we said, well, I want to look at risk factors for cognitive impairment, so I need people to be age 65. So there's a inherent age bias in all of our work that usually in our studies, we're starting to look at people in their 60s. Now, there's also wonderful work going on that follows people from younger age groups. And we can use that information to inform what we know today. And when we think about what are the modifiable risk factors for Alzheimer's disease and related dementias, there are population-based trends in how we treat and screen for these risk factors. For example, hypertension is on the rise, but more people are getting treated. What does that mean today when we're looking at a group of people and we're looking at midlife hypertension? Or we're looking at hypertension that was from the 1960s where we didn't really do as much directed screening. At the same time, we are experiencing changes in life expectancy across different groups of our population. So how does that affect what we know about risk and resilience? When we recruit from a memory clinic, we get a lot of people that are worried well. We see differences in levels of education, and these differences could potentially end up with a study that may not represent the underlying structure of the population that we want to study. What happens when we use electronic health records or electronic medical records and we use one integrated healthcare delivery system? You can pre-specify people of a certain age or race or ethnic group. People have fairly equal access to care. They may not have equal usage of care. And it's a wonderful opportunity to understand differences in rates in a population that in theory has the same care and risk factor screening. So if you were, for example, to see a difference in rates of Alzheimer's disease in men versus women, you would say, well, we know at least it wasn't due to differences in care. Nonetheless, a very strong healthy volunteer effect, if one is building a cohort study and bringing people in for research visits from an electronic medical record integrated healthcare system, there's always people who volunteer first, non-Hispanic whites, women, and higher levels of education. You can also use a population sampling census-based approach. This requires a lot of resources, and the goal is to reflect the underlying population. But it might not work that well across all segments of a population. This approach is really, coming from the notion that everyone kind of feels the same and you're expecting the same recruitment rate across different groups, and we can't make that assumption. Grassroots, you have to establish trust and connections. If successful, you have increased representation. But you do have to also worry about a snowball effect. So there's sharing of an experience. So if friends of friends of friends sign up for a study, that can make it difficult if you have a blinded aspect and also requires resources. So just to remind everyone, we have this great publication from Dr. Hill that really hones in on, who are the priority populations. And we heard some great talks today on these populations and how important it is to engage them in our work going forward. And we're talking about, of course, different racial ethnic groups, but also people with lower levels of education, disadvantaged populations, and things like low literacy. When we're thinking about health disparities in our work, I want to remind that it is the unequal distribution of health and illness due to unfair economic arrangements, poor social policies, and bad politics, not caused by differences in genes. So this was work led by Dr. Elizabeth Rose Maeda that at the time was the first work that had looked at incidence of all-cause dementia using Kaiser data and showing very large differences over a almost 13-year period at rates of new cases of all-cause dementia. And it was the first time anyone had looked, for example, at Asian Americans as a whole, and this was in 2016. So you can look at that and know, well, this maybe wasn't due to differences in healthcare. So this is a hypothetical study that you did a study. You have about 1,000 people. 80% of the women in your study were recruited from memory clinic. 75% of men were from grassroots events. 90% of your non-Hispanic whites were from a college campus health education event. 60% of Asians were recruited from an electronic medical record mailing, and 70% of your non-Hispanic whites were recruited from a social media campaign. So sounds like a great study, right? You have men and women, you have people from different, racial ethnic groups, but what you have here is a ubiquitous bias in that these sampling methods worked for some populations and didn't work for others. And so when this happens, you can make conclusions from your study that represent overall a diverse group of people, but you cannot necessarily answer whether you have a disparity in the association between your exposure and your outcome. You can't say if there was a disparity due to male versus females when you have a ubiquitous bias in your sampling method. So this is work done by Dr. Paula Gil-Sanz, again looking at people in Northern California, but those who were born in a high stroke belt had a very elevated risk of all-cause dementia. And this is in people who already moved to California. And this is an example of we actually don't know the true impact in this study of living on a stroke belt on long-term risk because they had moved to California. So again, we have selection at every step of a study. This is looking at midlife cardiovascular risk factors in our Life After 90 study. So these are people who made it to age 90, but in midlife, quite a lot of them have high rates of cardiovascular risk factors. And we're now going back and looking at everyone who would be alive and would be in their 90s today, and how does this differ from those, and how does this differ from trends in the population at that time with those risk factors? So inclusion is a science of itself. We have to acknowledge our history, trust. It takes time and resources. We have to think about how we reach out to different communities. Requiring people to come to the ivory tower does not work. And we have to encourage in our participants a return on investment. And I'm going to show a few slides of a brand new study called GROW. That's the Greater Sacramento Area Women's Brain Health Program. It is both a program and a study. And co-investigators are Dr. Wayne Meyer and Kristen George from UC Davis. And we want to engage women from historically underrepresented communities by cultivating long-term partnerships with the community and providing the opportunity to engage in research. Our goal is to enroll 700 women. What was very interesting about this study is due to an IRB delay, we were in the community for 14 months doing brain health education work, working very closely with our community advisory board, which Dr. Wayne Meyer is doing a fantastic job of, and delivering free brain health education that is customizable. We do different things at health fairs, community and cultural events. We also do some exercise classes. This is just free and open to everyone. We have a number of events per month. I think I accidentally said last night to someone we have 30 events per month. No, we've had 37 events over the last 3 months, but we have several events per week. And in 3.5 months, we have seen 94 women. And the 94 women who you can see, not surprisingly, are non-Hispanic whites, are making up 43%. But we have a lot of people in these other groups as well. And you can see in the percentage that are scheduled that our demographic is shifting. So if you are a non-Hispanic white woman, there is a 6 to 12-month wait now to be in our study. But what's unbelievable is that 100% of these participants so far are being recruited from grassroots. We have a plan for EMR or perhaps social media in the future. We may not be considering that at all, and this may end up being a 100% grassroots study. So it's been giving us an opportunity to actually understand the science of what works, and what works is to be in the community for a long time. And we have real-time tracking of our demographics and different curriculum. We are building a community network database, so we're actually analyzing what worked and for whom and for which community. And this is a very powerful tool that I hope to extend out to our other work at the ADRC and our other studies. So is it simply enough to increase inclusion? No. We have to contextualize our research, demonstrate return to the community, appropriate engagement, compensation, and access. I think our network really has the potential to be a leader in uncovering new risk and resilience factors for these different groups, but through intentional approaches to reduce selection and sampling bias. And so if we want to really increase representation, we have to think about what it means. Who do we want to be representing? And we have to think about our selection bias at every phase of the research. So this has to be done in tandem with recruitment science, or else we can't be successful. Underserved is a broad term, and it really captures everyone who's not already part of ADRC research. And I had a slide, and I'm sure I left other really important groups out. And this is something we need to always be thinking about. Who are we not including in our work? Thank you.
Thank you, Dr. Whitmer. It is a privilege to invite and introduce our next speaker, Dr. Robert Califf. Dr. Califf is the FDA Commissioner. As the top FDA official, he's committed to strengthening programs and policies that enable the agency to protect and promote public health. Prior to rejoining the FDA, Dr. Califf was head of medical strategy and senior advisor at Alphabet, Inc., contributing to strategy and policy for its health subsidiaries, the Verily Life Sciences and Google Health. In the past, Dr. Califf has served as Vice Chancellor for Clinical and Translational Research at Duke University. He has also served as Director of the Duke Translational Medicine Institute and was the founding director of the Duke Clinical Research Institute. Dr. Califf is a cardiologist and an internationally renowned expert in cardiovascular medicine, health outcomes research, healthcare quality, and clinical research. He has led many landmark clinical trials and is among the most frequently cited authors in biomedical sciences, having published over 1,300 papers. Dr. Califf is a member of the National Academy of Medicine and has served on the NIA Council as well as on the advisory committee for the NCI, NHLBI, and the National Institute of Environmental Health Sciences. It is a privilege to invite Dr. Califf to make his presentation on real-world data with a focus on electronic health records. Dr. Califf.
Yeah. Thanks so much. Can you hear me okay?
Yes, we can hear you. Thank you.
Great, great. I really appreciate the chance to be with you all. And sorry I can't be there in person, but let me tell you, your mission is very much on my mind right now. I spent yesterday with a very close relative who has advanced dementia and is really having trouble getting through life in a so-called memory facility. And the strain on the family and everyone involved is really something to behold and a very important thing for all of us to work on. I'm going to be brief in my comments because I think the Q&A would be the most important part of this, but just make a few points in general about real-world data and the way we think about it from the perspective of the FDA. Just having heard the thoroughness of the previous presentation, mine will be a very broad set of comments, but maybe in the Q&A we can focus down on the common ground and the issues that need to be addressed in this very quickly evolving arena for clinical and translational research. First, just to make a point about the perspective of the FDA in this, while my career has been spent in all the different parts of research, I'll comment today just from the perspective of FDA, which of course is looking at medical products. By the way, also looking at food, which may be an important component of this as we come to understand that better. But for now, let's focus on medical products, either diagnostic tests, very important in this field, or therapeutic interventions, drugs, devices, biologics, all of which are important in this field. In general, the law defines what the FDA can do. And I think it's very useful when we talk about electronic health record data, real-world evidence, to in general divide the world into early phase studies, phase 1, phase 2, many phase 3 studies, where it's really not appropriate to talk about real-world data because the real purpose of the early phase studies is to weed out the interventions and tests which are not going to meet the standards for safety and effectiveness. And it's an important point that even as of today, despite all the tremendous improvements in science that have been made, it's still the case that about 90% of drugs that start phase 1 evaluation in patients, that is, in the clinic, about 90% of those interventions turn out to have risks that outweigh the benefits, or may engage the interesting biological target, but not have the clinical effects that were expected, and so don't make it to market. And so that kind of research, the early phase studies, needs to be very focused, very specific, and for the most part separated from clinical practice, because intensive measurements need to be made for obvious reasons. When you're in a situation where 90% of the time the risks are going to outweigh the benefits, or there will not be sufficient benefit to make it worthwhile, it's really critical that we only enter people into those clinical trials to the extent necessary to get the answer. But then beyond that point, things really open up and become quite different. And this is been a critical discussion that has occurred within the FDA and also between the FDA and the rest of the ecosystem that deals with evaluation of medical products and use of medical products in practice. Once we get beyond that phase, the FDA has very little control once a product is on the market, remembering that the mandate by law that we have is that the product needs to be shown to be safe and effective for an indication in a particular clinical setting that can be described in a package insert. And then beyond that, the use of the product is an issue of medical practice, and the FDA does not regulate medical practice. The caveat here is in a situation in which there's a particular safety problem or where accelerated approval is used, then the FDA has authority to require particular studies. But for the most part, when we're talking about real-world data, real-world evidence, we're talking about an ecosystem which is well beyond just the FDA. The FDA has a role, but also a critical role for NIH, for CDC in circumstances, and increasing interest by CMS because the issue with the IRA of payment over time for interventions is a very big issue that we're all concerned about and want to understand the best way to reimburse for effective interventions, but also reduce the use of ineffective interventions in people in whom the benefits are not going to be realized. So that's sort of a comment about the ecosystem and the setting in which we operate. And I'd say there's a tremendous effort going on right now across at least the federal landscape to work with the private sector in the post-market space to basically generate more evidence that can be used in practice so that the right people get the right treatments at the right time, and people who do not stand to benefit are not exposed to interventions that are going to be ineffective or have toxicities that outweigh the potential benefits. Now, having said that, it's very important, I believe, that when we talk about real-world data and real-world evidence, to realize that we're talking about two related constructs, but they're not the same thing. In my career, I'm a cardiologist. I've spent the early part of my career developing interventions for heart attack and unstable angina and heart failure. And in those conditions, real-world data was a standard part of what we use. That is, people coming into emergency medical settings, there's not time to say, why don't you go to the research clinic next door tomorrow? You have to enroll the person in the study and collect the data from a variety of sources, many of which are coming directly from electronic health records or abbreviated forms that are collected in the course of routine practice. So real-world data increasingly now will refer to electronic health records. The great thing now is all Americans have an electronic health record, essentially. We all have claims data, which is increasingly well curated. And the science of understanding how to collect the data once and use it for multiple purposes is moving along very quickly. But what's often underestimated is that to take real-world data and derive real-world evidence, that is a conclusion about the effect of, let's say, an intervention on the outcomes of patients, that requires a method applied to the real-world data, a study design, and a knowledge of the appropriate statistical methods to derive the appropriate conclusions. So real-world evidence can be derived from real-world data, but just using real-world data in no way gives you the evidence that you need to draw the conclusions that might be most important. And in this regard, many of us believe that while there's tremendous value in real-world data and observational analysis of that data to derive conclusions, The most underused method is randomization done in the real world so that we can draw a causal inference about whether a treatment has the effect that we hope it'll have, or that we can discern, for example, toxicities and make sure that things that may occur after someone takes a treatment are due to the treatment and not due to the natural course of disease. And so this distinction between real world data and real world evidence, and the idea that real world evidence means not randomized, is something that we very much want to change into thinking that as we get better and better with the quality of electronic health records and claims data, and we now add in things like sensors and the use of assessments done through the cell phone, all sorts of ways we can now derive data from the real world. It's very important that we also think about randomization as a tool that can be used in that setting. Also, I want to just make a comment about the view that AI is going to make a big difference here. And I think it really will, because if we begin to think about how to knit previously disjointed elements of real world data together into a coherent picture, the use of artificial intelligence applied to electronic health records, I believe, is going to be revolutionary. And we're going to need to make the best of it from the point of view of deriving research data from this improved ability to use things like clinical notes and conversations to derive the kinds of data that can then be analyzed in a way that will enable us to derive conclusions. So as I think about the disease that you all are dealing with in the midst of course, in this job, it's kind of a little dizzying to think about all the different things that I deal with in a day and try to understand. There is no question, in my view, that if we look at the course of a person over decades who may be at risk of Alzheimer's or may benefit from a preventive intervention, it's going to be absolutely critical that we advance the use of electronic health records, that we get better and better at curating the data that's there, and that we also develop standards and common terminology so that we can work across health systems and follow people over long periods of time to understand the long-term impact of the interventions that we're using. And so, in conclusion, hopefully getting us back to on time here, I just want to really leave the view for the group that there's a keen focus on this across the federal government. My previous work in big tech and in academic medicine also leads me to be very optimistic about the likelihood that public-private partnerships here, and particularly involving activated patient groups, can make a big difference in moving this along faster so that we can sort through potential interventions much more quickly. And also, as we put things on the market through accelerated approvals and other mechanisms, assure ourselves and everyone else that we're measuring what happens with these interventions so that we can do more of the things that work and less of the things that don't work. So I'll stop there and look forward to any questions or comments.
Thank you, Dr. Califf. Any questions, comments, anyone? Any comments, questions, anyone? Yeah, go please.
Robert, thank you very much. This is Eric Ryman from the Arizona ADRC. Speaking as a prevention researcher, to follow your comments, imagine we had a prevention therapy that showed a clinical benefit in the next few years, but despite our best efforts, we haven't had a sufficient number of individuals from underrepresented groups to confirm the benefits and risks in those individuals, or maybe even we had an accelerated mechanism to get approval soon. Could you envision the opportunity to use real-world data post-marketing, perhaps with a randomized staggered start approach, to be able to make that treatment available now and without waiting many years for people to be on placebo to get additional evidence of a benefit to extend its use to a broader group of individuals?
Eric, I haven't seen you in a while. It's good I can actually see you here, so that's good. Good to see you're still standing. That's impressive. I mean, the short answer is yes. And I would argue, if you go, maybe I got spoiled in my early career because as we were figuring out what to do with heart attacks, we were randomizing 10 to 40,000 people around the world routinely. And then after an intervention got on the market, there were multiple observational studies and clinical trials that were done. I would argue that because in these kinds of interventions, it's highly likely that there will be heterogeneity of treatment effect and also heterogeneity of risk profile, creating a different balance in different kinds of people. One dimension, of course, is the issue of underrepresented minorities, but you could also imagine that duration of disease, age, renal function, all these characteristics that, in an initial registration trial done rigorously, you're not going to have all those characteristics represented. And I think the way out of that box is the use of real world data, as I say, where possible, adding randomization. You might imagine, and we did this in a lot of areas of cardiology, working at the margins where you finish the clinical trial and you're really just not sure, and enough so that the clinical community really has equipoise. And I think for the therapeutic antibodies and that we're dealing with right now, that's the case in a lot of populations. There's still a strong argument made for randomization, perhaps adaptive, as you say, or using a step wedge kind of approach, even in populations. I think in terms of how this relates to labeling at the FDA, I think you'll see increasing openness at FDA, but it's really more a matter at that point of the clinical community and how it wants to adopt these therapies based on evidence. But the short answer would be yes.
Any other questions, comments, anyone? Dr. Califf, I'm wondering, in terms of real-world data, we get data from all kinds of healthcare systems across the country. And the issue of integrating data from different healthcare systems who may collect different variables and may have differing expertise in diagnose something like dementia, which is a complex disease to diagnose, curate those data. Would love to hear your thoughts, how you think those issues could be dealt with.
You just said a mouthful there, so I'll riff on it a little bit. And I've been careful to not say too much because I could talk for hours about what you just raised. At the very basic level, I feel like right now, most of our efforts using real world data need to focus on exploratory observations that lead to subsequent, more rigorous trials or detailed epidemiologic studies, or can be constructed to look at the simple things. Like there are big questions related to big things that happen to people that are not so hard to measure, like does a treatment lead to more or less hospitalization and death, something that we're getting better and better at measuring. But even there, we have major issues across health systems. If I didn't know better, I would think that the American health system was set up on purpose to create a fragmented, piecemeal approach that makes it impossible to track people over time. In fact, that is what's happened. Whether it was on purpose or not, we could have a discussion about, but you're well aware, and the last speaker referred to it. Some of the most important information, of course, is in the people that are missing the follow-up or who change where they get their health care. And right now we have a system that makes it very hard. We can do it with contortions, using probabilistic matching in follow-up from various sources. But for the kinds of information we need to construct a system that enables follow-up of people over time. And this is a focus of our discussions at the federal level. Even something as simple as knowing who's dead and alive. It surprises people when I say we don't know who's dead in most electronic health record system. You'd think that would be known. The second level, I think, is for the simple things, standard terminology and data standards. And I would say a lot of progress is being made there in the ability to curate using common data standards from data that may not have been collected totally using common standards. But the more that we can work with Office of the National Coordinator, for example, on national standards through USCDI and the research additions that are being made to that, the better off we're all going to be. But then you get into the area where I spent a lot of time, which is how do we, when we have very specialized information, and for you as in your community, it's really the assessment of function that's so important. For me as a cardiologist, it was often what are the details of the coronary anatomy, or it wasn't was an echo done. It was what did the echo show about mitral regurgitation, so on and so forth. I think that is something where the community of specialists is going to need to work at sort of two levels. The first is what are the derived elements that can be done in the real world without destroying the ability of someone to practice. And then the second is for the more rarefied things that you use routinely as specialists, how do we have hybrid studies that take advantage of the real world data where it's useful, but then add in the more detailed measurements? And I think we're beginning to see increasing numbers of studies that are able to do that. A good example would be randomization within registries that now is becoming more commonplace in my specialty of cardiology. But all those tiers of things need to be considered as we work through this. And I'll just, the last thing I'll say, I think we're in a situation now, you referred to the difficulty of people who are in practice being able to participate. We're in this very critical time, I think, now in clinical practice where clinicians are under assault, basically, or they feel they're under assault. And as I've gone around talking to people about real-world data and how we can be helpful at the FDA, the comment I get most commonly from clinicians is we're being beaten up to generate as many RVUs as we possibly can. How can you expect us to do anything in addition? But what's really interesting now is I'm 100% sure that generative AI is going to make it possible now to see patients in a clinic the way we used to do it, where you're actually looking directly at the patient and the family and having a conversation. And you won't be spending all your time clicking boxes in Epic to make sure the billing is correct. And the question that's before us is, are we running a health system in the US or a healthcare enterprise where when that happens, clinicians are going to be told to just see twice as many patients in the same period of time? Or are we going to be in a system where that extra time is used to have the discussions with patients and to take the time to get into a learning system where the data are collected as part of practice and learning goes on continuously. I think it's pretty obvious, which I'm hoping for, but the signs right now are not too optimistic. So I hope people like you will be very vocal. I was just up at Mass General Brigham. I never did any training there, and I think Duke was a great place, but I have to admit it's sort of the pinnacle of academic medicine right now. And when you look at what's happening there, talking to clinicians as I did during my visit, there's a real worry that the clinicians are just financial objects now to be manipulated in a system that's financially run. I'm not talking about MGB specifically. I'm just talking about the general situation that we're in the U.S. So I'm expressing frustration. I think the tools are coming into place. The technology is here, and smart people like you know what needs to be done. But we're going to have to look and recreate our health system to deliver the goods. And I'll just close by, the observation is we're inventing all the new things that the rest of the world depends on. If you look at, as I go around the world in my international role, people are very jealous of FDA and the U.S. discovery enterprise. But then we're almost in last place among high-income countries in patient outcomes. And so something's wrong. And I think this issue of how we learn and practice and apply it is sort of one way to look at what's wrong and try to fix it.
Thank you so much, Dr. Califf. We really appreciate your time and the knowledge you shared with us. Thank you so much. We are very grateful. Thank you.
Thanks for all you're doing, and take care. Have a good rest of the meeting.